Shay uses a portable water bath to collect the cryoglobulin sample from the patient, who is freshly fed by a nurse. A temperature of 37.5 degrees is maintained throughout the process of separating the serum from the blood to prevent the precipitation of the proteins. Shay is spinning the sample in the centrifuge, which is set at 37.5 degrees. He then pours the serum into a 15ml tube to be stored in the fridge. After 7 days, the precipitate has been allowed to form. Mira spins the serum in the centrifuge at 4 degrees, maintaining the cold temperature. Schaff then confirms the presence of the precipitate by the appearance of a white pellet. The pellet is then washed 3 times in cold PBS. Amira is using the hydrosys to carry out the procedure called urine fixation, which is performed on urine samples as well as cryoglobulin samples. The washed proteins are redissolved in the water bath at 37.5 degrees and resuspended in the solution using a vortex mixer. Amira dispenses the cryoglobulin sample onto the applicator combs along with an in-house control. She transfers the applicator combs into the humidity chamber to allow the samples to diffuse while she preps. She removes the excess liquid from the gel and places it onto the analyzer, followed by the buffy strips. Finally, the combs are placed on the analyzer. Once the proteins have migrated along the gel surface, a standard mask is used to apply the antisera, which binds to the specific proteins. Here, Amira is dispensing fixative antisera, anti-IgG antisera, anti-IgA, anti-IgM, anti-kappa, and anti-lambda antisera. This is incubated for 5 minutes, and then any excess antisera is removed using thick filter paper. The gel is then dried and finely stained, ready to be read. Here Steve is reading and reporting the gel. The patient tested negative for the presence of cryoglobulin protein, as there were no bands present on the gel. Cryoglobulins are proteins that become insoluble in blood when the body temperature drops, causing blood plasma to thicken, which may result in blocked blood flow to tissues and organs. Here is an example of a patient positive for cryoglobulin.
A quantilizer is an analyzer that carries out a technique called indirect immunofluorescence. This is used to look for antibodies in a patient's serum to see if they have an autoimmune disease. The process is completely automated once M has loaded the consumable samples and reagent. M loads microscope slides, which have on them a thin layer of rodent tissue. This tissue contains the target molecules that will allow us to detect the antibodies in the patient's serum. The samples are pre-diluted with saline solution in a series of dilution wells. Saline is utilised instead of water to prevent osmolysis. This is a rupture of cell walls by excess water. Now the slides are being loaded with the positive and negative controls, followed by the patient samples. The incubation period takes 20 minutes, and this allows the antibodies, if present in the patient's serum, to bind to the target molecules in the rodent tissue. The unbound antibodies are washed off the tissue before a fluorescent marker is applied. This is then incubated for a further 20 minutes, and the marker binds to any patient antibody on the tissue so that they can be seen when looked at under the microscope. Any unbound marker is then washed off before M covers each slide with a cover slip, ready to be read under the microscope by Tina. A liver autoantibody screen is requested in patients with liver function issues and could be due to a disease called autoimmune hepatitis, which if left untreated can lead to liver cirrhosis and ultimately liver failure. Positive samples containing patient antibody will give off patterns of apple green fluorescence on the rodent tissue. Negative samples will appear dark under the microscope.
HLA-B27 is a blood test used for a surface protein on white blood cells, which help the body's immune system differentiate between its own and foreign cells. Duroclone B27 reagent is used to incubate patient whole blood. The red cells are then broken down using lysing solution. Sandeep is checking to see if the red cells are completely lysed and if the sample is ready for analysis. The assay uses target fluorescence channels to determine if cells are B27 positive. HLA-B27 positive results are associated with an increased risk of developing certain autoimmune disease. Flow cytometric HLA-B27 screening is rapid and cost-effective. The test is often performed due to its association with ankylosing spondylitis, which is a long-term condition in which the spine and other areas of the body become inflamed.